Hi, my name is Alison Moskowitz, and I'm a lymphoma specialist from Sloan Kettering, and I'm going to be talking about novel agents in Hodgkin lymphoma. So over the past 5 years, we've seen the approval of two very active drugs for Hodgkin lymphoma. In 2011, brentuximab vedotin was approved, and nivolumab was just approved in 2016. So I think now our current challenge for Hodgkin lymphoma with regard to novel agents is how do we incorporate these very effective and often fairly well tolerated agents into our current treatment paradigms, and also where do the other novel agents fit in. Brentuximab vedotin is currently undergoing evaluation in multiple different clinical trials, and including studies in the frontline setting, and I think we'll be seeing the results of those soon. And with regard to the other novel agents, I think it's important to think about them in the context of how do they impact anti-tumor immunity. This is in particular due to the success of anti-PD-1 therapy in Hodgkin lymphoma, nivolumab and pembrolizumab, which have shown to have significant activity in Hodgkin lymphoma, leading to the approval of nivolumab. And in the cycle of anti-tumor immunity, many of the other novel agents impact various other steps of this process and are likely to complement the activity of nivolumab or pembrolizumab. These agents include HDAC inhibitors, lenalidomide, inhibitors of PI3 kinase pathway, as well as JAK-STAT pathway. With regard to an HDAC inhibitor, Mocetinostat has been evaluated in Hodgkin lymphoma and have been associated with response rates in about 1/3 of the patients. And an interesting finding with regard to Mocetinostat is that it's associated with upregulation of OX40 ligand in Hodgkin lymphoma cell lines, likely implicating that it has an impact on anti-tumor immunity or development of tumor-specific T cells, and therefore can impact anti-tumor immunity. With regard to lenalidomide, it also has activity in Hodgkin lymphoma, has been associated with a response rate of only about 19%, although clinical efficacy overall, or clinical benefit in about 1/3 of patients who receive lenalidomide. And it also has immune modulatory effects likely in Hodgkin lymphoma. One of these effects is likely through proliferation of T cells, and also in particular the development of tumor-specific T cells. Everolimus, an mTOR inhibitor, has been shown to have activity. This was evaluated in Hodgkin lymphoma based upon the activity of the PI3 kinase mTOR pathway in Hodgkin lymphoma. And in 2 different phase 2 studies, the response rate to Everolimus was found to be in the range of the 40% range. And Everolimus likely has an impact on anti-tumor immunity, potentially through development of CD8 positive memory cells. And so it's potential that it can enhance the anti-tumor immunity in Hodgkin lymphoma. And finally, JAK2 is a potential important target in Hodgkin lymphoma, and this is particularly given the commonly seen amplification of the 9p24 chromosome, which contains the genes for JAK2 as well as PD-L1. Overexpression of JAK2 leads to increased expression of PD-L1 on Hodgkin lymphoma cells, and therefore inhibition of JAK2 would be expected to downregulate PD-L1 and therefore, again, impact anti-tumor immunity. There's very limited data with regard to activity of JAK2 inhibitors in Hodgkin lymphoma. This was a phase 1 study that enrolled various different histologies and included 12 patients with Hodgkin lymphoma, 4 of which had some degree of tumor reduction. There is an ongoing phase 2 study with ruxolitinib, which is a JAK1/2 inhibitor in Hodgkin lymphoma that is led out of Cologne University. So putting these agents all together and how they impact the steps involved in anti-tumor immunity, we have nivolumab and pembrolizumab, which impact the direct activity of tumor-specific T cells and killing of the tumor cells. Brentuximab and bendamustine, as well as idelalisib, through directly killing tumor cells, likely lead to release of antigens and further stimulation of an immune response. JAK2 inhibitors both downregulate PD-L1 as well as impact dendritic cell maturation and therefore can impact the antigen-presenting cells. And finally, lenalidomide as well as HDAC inhibitors and mTOR inhibitors impact the T cell priming and activation and proliferation and the quality of the immune response. And when we're thinking about putting these drugs together, we know that the anti-PD-1 drugs such as nivolumab and pembrolizumab are very active in Hodgkin lymphoma. However, the complete response rates are fairly low. And so it's possible that by impacting other aspects of the anti-tumor immune response through using some of these other agents that we can improve the activity and particularly improve the complete response rate. And so we will likely be looking at many of these combinations going forward. Thank you very much for your attention.